Good day, this is the Professopher, drwillphd, PhD, and today's Thursday, January 12th, 2023. It is 5:26 and 2 seconds, AM Eastern Standard Time, and today's research, cancer cell migration. So let's hop to it here. The physicist models cell migration to learn how cancer cells navigate tissue. Date January 11th. 2023. Source: Virginia Tech. And this cell migration poem is one of the ways cancer cells navigate tissue during metastasis. In a new study, researchers have explored mesenchymal migration through cell simulations and mathematical modeling. And their aim to learn more about how cancer cells size up surrounding tissue, pore stiffness, and adapt their gecko-like movements in response. As before choosing this mode of movement, the cell sizes up the surface to which it may stick. If the surface isn't too stiff or soft, and the path forward isn't too constricting, the round cell will rapidly grow protrusions that act like temporary limbs, jutting forward and sticking to the surface. The cell will then pull itself forward and retract its rear and repeat the process. The model and its insights on mesenchymal migration are a first step in learning how cancer cells migrate on the whole. Cell migration is complex. The cells make use of multiple modes of migration, both individually and in colonies. That is the advantage of coming up with a computational model. We can dissect the effects of many ingredients at play. The cells can grow and attach their protrusions efficiently to stiff substrates, and if cells grab onto too soft tissue, they'll end up putting it back toward their bodies rather than using it to pull themselves forward. And their simulations confirm to the researchers that cells distinguish between soft and stiff surfaces by comparing them to the physical properties of their own soft bodies. Substrate material properties will then affect the directions cells take as well as how efficiently they move. And to ensure that the model accurately simulated cancer cell migration, the researchers built in not only how cells respond to the substrate mechanics of tissue, but also how they tune their internal biochemical signals. And while navigating tissue, cells may also respond chemically to the secretions of a nutrition source in the body. The researchers' model is the first to simulate how both of these drivers of cell motion play out. The researchers found that cells prefer moving in a direction determined by their strong internal chemical signaling, whether or not the overall motion is efficient, but without a strong chemical signal to follow, they focus on substrate properties. And by piecing these elements of mesenchymal migration together and reproducing them in a model, researchers see a move toward better understanding and pinpointing how and where metastasis may occur. Metastasis also may involve multiple cell migration modes. Mesenchymal migration tends to be the initial mode of migration through tissue and into vessels, but cells often pivot to amoeboid migration. The researchers say, whereas cells move like geckos in the former mode, the latter has them moving more like tank threads. As chemotherapy works well against cancer cells and mesenchymal migration, but not as well when the cells switch to amoeboid migration. For experimentalists to understand that transition, they first need a better grasp of the mesenchymal mode. The researchers hope to use a model to look how cell-cell interactions may affect migration as individual cells bump into one another and set off changes in their direction. They also want to learn how cells negotiate more curved, narrow channels in their microenvironment. As the researchers investigate each effort to more closely model cell migration brings the team closer to understanding how cancer cells invade the body. The researchers want to come up with a predictive model that can produce new types of qualitative behaviors to explain more measurements and motivate new experiments. The experiments are quite comprehensive, but they significantly benefit from simulations. For instance, when it comes to resolving very small timescales in the dynamics of these hill deformations, they are basically discerning all those components. And this is the Professopher, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.